Hello, my name is Professor Matthew Schmidt, and I'd like to welcome you to Genetics. As you can see, uh, this slide is entitled Gene Expression and Protein Synthesis, and that's what the thrust of the next several videos are going to be about, the next several sessions. So we're going to talk about this. Well, let's talk about it right now, but we have to start somewhere. We're really getting into molecular biology, and not just from the point of view of DNA, from the point of view of how the information encoded in DNA actually is manifested in an organism or a cell. So we're starting in this session with transcription, RNA, and the central dogma. These are all related topics, but before we start, let's do something that we've done before, and that is to sort of see where we are. I don't necessarily mean in the course, although that's partially what I mean, but I mean where historically do we stand in terms of our understanding based on everything that we've thought about so far. So we have this idea hopefully ingrained in our heads that structure and function are very importantly going together, but that there are different ways of getting at one versus the other. So we've got now the structure of DNA down, courtesy of Watson and Crick. We understand, courtesy of them as well as Maselson and Stahl and others, how it can replicate, and we also understand the basic idea of how it can store information and allow variation. If you remember, replication, storage, and variation were the three things that we said needed to be able to be done by the molecule that, DNA that was going to be the genetic material. But we are at a point where we don't specifically understand how the information in DNA is used to determine phenotype and how mechanistically that actually occurs. What we do know, because functionally, remember, starting with Beadle and Tatum and Ingram we talked about, we know that somehow, remember the one gene, one enzyme idea, we may have modified it a bit so that it was one gene, one polypeptide. But the bottom line is we do know from that and putting all of this together that somehow the information in DNA is used to make a polypeptide that has a specific function. So we want to start to really unravel how this occurs. Now, we haven't spoken a lot about RNA yet, but this is the time to do it. So a lot of things, after Watson and Crick published their paper, things started to move really, really quickly. So in the 1950s, not too long after they published their paper, an important discovery was made. Now, the initial experiments were done, so many of these initial experiments were done in E. coli, in bacteria. But we're not going to go into the details of this experiment, but suffice it to say that some experiments with bacteriophages in E. coli implied that RNA did have a role as an intermediate in the process of gene expression, which really means gene expression is using the information in DNA to make a polypeptide. Usually that's what it means anyway. I want to look, again, we're not really going to go into the experiment, but logically, even though that experiment was important initially, I think you'll be able to relate to this one a little bit more, because it was also found that in eukaryotic cells, so of course the one big difference between eukaryotes and prokaryotes is the fact that the DNA is sequestered in the nucleus of a eukaryotic cell, whereas the protein-making machinery, the ribosomes, are outside the nucleus, right? So what they showed was if you radioactively labeled RNA nucleotides and sort of literally followed where in the cell they went, over time they first collected in the nucleus, the idea being around where the DNA is, and then they literally migrate out of the nucleus into the cytoplasm. So once that was determined, there's a very strong implication that RNA acts in some way or another as an information intermediary or intermediate. In other words, somewhere between the DNA and the polypeptide, we're now getting another step in there that involves RNA. Now again, biochemists knew RNA existed just like they knew DNA existed before its significance was fully known. So now it's time to understand RNA's significance. And it turns out, no offense to DNA, that RNA is a lot more of a versatile and perhaps interesting molecule. You can decide. So Francis Crick of Watson and Crick, Watson went on to do a lot of important things, but Crick sort of stayed more in this path of molecular biology and at least was a part of most of the major discoveries about how this all worked. And Crick coined a term to explain the basic flow of information in a cell. If you actually can find some of the papers that Crick originally wrote, you might find them interesting. We're just going to be, you know, jumping to the conclusion, but he explains exactly why he did it this way. So he came up with an idea called the central dogma of molecular biology. Um, just for the record, the word dogma, usually some people said this is a bit odd. Dogma in religion means sort of something this is the way it is and it's not to be questioned, I think. And Crick was asked about this later, and he said something like, I don't think I really knew what that word meant. He just meant that it was a strong hypothesis or a basic rule type of a thing. And it is a basic rule. It's not to say that this can never, ever, ever be, I should say it a different way. It's not to say that things must happen this way all of the time and there are no exceptions. But it is a pretty darn good rule that applies most of the time in normal situations. So what he basically said was that DNA is where the initial information resides. RNA is an information intermediate. If we're talking about a eukaryotic cell, which actually he wasn't necessarily, but I wrote here, brings the information from the nucleus to the cytoplasm. Even if you were in a bacterial cell, though, this journey doesn't have to be made, but the RNA is still the transmitter, the messenger. You've probably heard of messenger RNA, from the DNA to the place where the protein is going to be made. And then ultimately, the information in that RNA is going to be used to synthesize a protein. More technically, a polypeptide, but this is the idea. And we have a real fancy picture. There's a simple picture. But the central dogma, and sometimes you'll see it in different forms, and Crick had some very specific reasons why he did it the way he did. We don't need to dwell on that, interesting as it is. But basically, at the risk of being redundant, because this is so central and so vitally important, the basic idea is that in terms of gene expression, which is really what we're thinking about now, right, and protein synthesis. And by the way, the term protein synthesis, that's not a very good bracket there, but it usually means the entire process of getting from DNA to the protein, all right? But specifically, what the central dogma says is that DNA, I should say that DNA is used, and the information in DNA is used to make a messenger RNA copy. We'll talk about different types of RNA, but for now, let's think about messenger RNA. And the process by which that's accomplished is known as transcription. Then the information that's in that messenger RNA ultimately is used by the ribosome and other actors to create a protein, and translation is the specific process by which that is accomplished. Now, it really isn't part of the information flow, but many books will put in this loop over here, which is showing that you could almost think of it as dual function, because what I mean is DNA in the most fundamental sense really has two roles. One of it is within an organism or a cell to direct the expression of information into something tangible, that is a protein, but also it's the molecule of heredity. I'm not saying these things are totally separate, but when cells divide or when organisms reproduce, DNA has to be able to make a copy of itself, and we know that, right? So what's meant to be depicted here are the major ways that information flows, and going around like this means that basically DNA can make a copy of itself and make more DNA. DNA can also make RNA. DNA can't directly make protein. It does it indirectly, right? Messenger RNA is what's directly making the protein. So that central dogma is conceptually very, very, very important. And conceptual is great but we have to really explore at the molecular level what's going on in order to allow this to be true. Okay? Before we can do that, we have to say a few things about the biochemistry and properties of RNA. You may already know this stuff, but we haven't said it, at least not specifically. So RNA is a nucleic acid, just like DNA, and it is a polymer of nucleotides, but they're RNA nucleotides. You may or may not remember, I think we said what the difference is between a DNA nucleotide and an RNA nucleotide, and the major difference is, let's look at how RNA nucleotides differ from DNA nucleotides. Fundamentally, there's a couple of ways of saying this, but I just changed my mind about how I wanted to say it. One way of saying it is the reason it's RNA is because RNA nucleotides contain the sugar ribose, whereas DNA or deoxyribonucleotides contain the sugar deoxyribose. But what that really means, you guys, is that in ribose, or in an RNA nucleotide, the sugar at the 2' position has an OH group attached to it. In an RNA nucleotide on ribose, the sugar ribose at the 2' position has an OH group. If I had more confidence in my drawing skills, I would draw it, but I'm just going to draw a little piece of a nucleotide. Actually, a piece of the sugar. There's the 1' and the 2' positions. So in RNA, there'd be an OH group there. And I apologize if this is redundant, but when you deoxy something, all you're doing, you're taking away that oxygen, and what's left is a hydrogen. So in DNA, all there is a hydrogen at that 2' position. So that's important for a variety of reasons, but a lot of people don't think of it, that the deoxy means that you're basically taking a ribose or a ribonucleotide, taking off that oxygen. Now you got a deoxy. All right, so that's very important. If we skip ahead just 1 second to a picture of a piece of an RNA molecule, I guess I didn't have to draw. So here we have the base attached to the 1'. It's not drawn here, and the 2 is sort of going over the ribose there, but in RNA there is a hydroxyl group, an OH group there. In DNA you would just see the H. So that's one major difference. Another thing is that we've discussed the DNA bases. The RNA bases are similar, but there's no T or thymine in RNA. It's replaced by a very similar looking base, but not identical, uracil, U. Now, when it's relevant, and we'll see many cases where it is, U is complementary to A, just like T is complementary to A, adenine. So the 4 triphosphate nucleotides in RNA are ATP, GTP, CTP, and UTP. And you may remember we said that the names change, so the base thymine, for example, when you put it into a nucleotide, it becomes thymidine triphosphate if it's a triphosphate like this. So the base uracil becomes uridine triphosphate. A final important difference that really relates more to the entire molecule is that RNA tends to be single-stranded. It's not always the case, but for our purposes, let's say for now that it is a single-stranded molecule, not double-stranded like DNA. So let's go on ahead back to this picture and just get a feel for the fact that here's one nucleotide, right? And I'm just circling all the nucleotides that are getting strung together in very much the same way that DNA nucleotides are polymerized through a phosphodiester bond. Remember we said once that, here's adenine. It's in a chain here, so there's only one phosphate group there. But it was in ATP before it got transferred into here. I just wanted to reiterate that the RNA nucleotide is ATP. If you want to really be specific and make sure, say you were talking about a DNA nucleotide, you could write DATP there. In this case, though, that's not the case, so this is not ATP that's drawn on the screen here, but the RNA nucleotide that donated that would be ATP. So these nucleotides are one after the other, and you're seeing A, U, G, and C. There's no order implied by that. We're just showing you an example of the 4 possible bases. But like we said, there is a 2' OH there, so that's one difference. Uracil is present instead of thymine, right? And also you can see here that this is single-stranded. And from a functional point of view, that's extremely important because in DNA, I like this little sort of more simplified version over here. In DNA, of course, what would you have? You'd have pairing. Now, there's no U in DNA, but if it were to pair, it would pair with A. G, C, C, G, right? In DNA, everyone has a partner and there's always base pairing because it's double-stranded. Well, again, that's not happening here. So in RNA, these nucleotides are available if you want to think of it that way. They're not interacting with anyone. They're free to interact with someone, and you'll see that they're going to do that eventually when it comes time for RNA to do its job, or at least the job that we're trying to get at right now. Okay, so we need to get the rationale of transcription, and we'll go a little bit into the mechanism as well. What it basically means, as you've gathered, is the copying of DNA information into an RNA molecule, which is going to serve as an intermediate. Now, conceptually, think about this, you guys. Transcription really is similar to DNA replication in a sense, but be careful, in a certain sense. And here is that sense. In replication, remember, you have template strands, and because of the base pairing rules, you're able to use a template to synthesize a new strand based on those rules, right? Well, that same principle is going on here, but there's one or more, but there's at least one very important difference, and that is that in the case of transcription, only one strand of the double helix is being used as a template. So we'll see a picture of this, but the other strand just gets out of the way. In DNA replication, both strands are acting as a template, each of them making a new sort of complementary part so that you have 2 full new molecules, right? But the DNA is now acting just to make an RNA molecule, so only half of it, if you will, needs to be made. One strand is going to act as the template. That's very important. Also, another thing you might want to keep in mind is that when you do DNA replication, you basically want to replicate the whole entire molecule. When you do transcription, you're looking to only copy what's a relatively small DNA sequence into this new RNA molecule. It's one gene's worth of information. So we have to know we're starting at a very definite place and we're stopping at a very definite place. More about that in a minute. If you think about the process, so just for the same reason that if you're doing DNA replication, there's a natural sort of, because of the base pairing rules, alignment of new nucleotides coming in, right? So RNA nucleotides will align on the DNA template according to the base pairing rules. Remember, in DNA replication, we need a DNA polymerase to seal that covalent bond to make it stable. It's probably not too shocking that we need an enzyme, in this case RNA polymerase, to do the same thing in this new RNA molecule. So the base pairing rules are basically the same except with the UA instead of TA. They'll come together, but RNA polymerase needs to polymerize the covalent bonds that hold this thing all together. And very importantly, remember this, we said it when we were talking about DNA replication, that RNA polymerase, unlike DNA polymerase, does not require a primer. Remember the enzyme DNA primase. We mentioned at the time that primase has a lot in common with RNA polymerase. So evolutionarily, they may be derived from the same molecule, but the point is that RNA polymerase is better than DNA polymerase. I'm half kidding, but it's able to just show up and start polymerizing. It doesn't need any type of priming, so you don't have this problem about having to get rid of a piece, refill it in, and so in a way it's a little bit more streamlined. So new chain can directly be initiated with RNA polymerase. So here's, in a minute we'll see a picture, but we want to make sure that we are going to be able to understand and appreciate what we're looking at. So the DNA template strand, this is a bit confusing to some people. It's also referred to as the non-coding strand, and that seems a little bit strange because the non-template strand is called the coding strand. So let me quickly explain what that means. Let's say here's a piece of DNA. I'm just going to randomly make up the bases. I'm hoping I'm doing it right. 5 prime, 3 prime. All right, so let's just say that the top strand up here, well, actually I would like the bottom one for reasons you'll see in a minute, to be the template. All right, so I'm going to keep going here. Let's just make it something easy. A, T, A, C. What's going to happen is this strand, I'm not even going to bother filling in the actual letters. It just gets out of the way. The template is remember, only one strand is going to be used. So the new RNA nucleotides are going to come in here, C, right? U, because of the base pairing rules. A, U, G. So let's just pretend this would be a tiny piece of RNA, but let's just pretend that's all that we wanted to transcribe. So basically, in what I've drawn in blue there, that would be the new RNA molecule. So let's think about this. This would be the template strand, right? And this would be, up here, the non-template strand. What is the non-template strand doing? It does absolutely nothing, in fact. So then why do we call it the coding strand? And here's the reason. Some of you might see. I'm going to write it in green. Even though it did nothing, the non-template or coding strand is going to have the same sequence as the newly made RNA, except for the whole thing with the U's, because it was complementary to the template strand too. Let me say that again. The whole reason this RNA is getting made the way it is because complementary bases are being added. They're complementary to the template strand, right? But this non-template strand, isn't that complementary to the template strand as well? So if we filled in the bases up here that would be there, we'd have C. That's not showing up too well. I apologize. Erase that. We'd have C, T, T, A, T, and C. My point being, the sequence of this non-template strand, I'm sorry, it got a little bit all sort of caught up in the slide, but the point is that the sequence of the non-template strand, with the exception of the U's, which you can figure out, is exactly the same as the new RNA. So that's why it's called the coding strand. If you want to know the sequence of the RNA, look at the coding strand. It's going to be the same. It didn't really do anything, but by virtue of being complementary to the template, it's going to have the same sequence. I hope that came out okay. I really do. All right, so the template is non-coding for the very reason that it's going to be the opposite or the complement of what it just made, right? Okay. Now, this is really interesting. We've defined the gene in a couple of ways so far. Originally, most simplistically, we said it's a unit of information that controls a trait. But here's a more modern sort of a definition, and there are even more modern ones. But we can say that it's a sequence of DNA that codes for a very specific and functional piece of RNA. All right? Usually a messenger RNA, but not always. So this is a more molecular way of looking at what a gene really is. Another thing that's interesting, remember Chargaff, Edwin Chargaff had come up with the idea that the amount of A and T should be the same in DNA and G and C. Watson and Crick, you may remember, made use of his ideas in trying to figure out their model. Chargaff's rule will definitely not apply to RNA, just for the record, and the reason is because it's not double-stranded, right? The amounts of the 4 bases can be anything, and you'll see problems to solve that will make use of this. They'll say something like, what if you saw a nucleic acid and you did a bulk analysis of it and you found out that there's, You could make up any number here, 10% A, 20% T, 30% C, and then 40% whatever's left. That would not be a DNA molecule. That would be an RNA molecule, because in DNA, you have to have this because of the base pairing rules, right? Once an RNA is made, it can have any base sequence it wants. I mean, of course, it's controlled by the DNA, but the pairing is not relevant here because it's single-stranded. Now, in bacteria and prokaryotes, there is one RNA polymerase. It consists of 4 subunits that associate with another subunit called the sigma factor. The sigma factor does something special and produces the holoenzyme. We're not getting deeply into this. Your class may or may not. But remember when we had the DNA polymerases, we said that a lot of times there were multiple polypeptides involved. Here, and I'm speaking about bacteria for a couple of reasons. One, I like them. 2, most of this was discovered in bacteria. And 3, sometimes we know more about what goes on in bacteria than we know about what goes on in eukaryotes. Eukaryotes are a little bit more complicated sometimes. But there's only one RNA polymerase, so now we can visualize what's going on. If you wanted the real simple version, guys, I'm not even being silly. It would be start, continue, and stop. I mean, that's the really, really most simple version of it that there is. Remember what we said before. The starting has to occur at a very definite place, and the stopping also has to occur at a very definite place, because you basically want to take out of a long, long, long piece of DNA, say one gene is right there, you only want to copy that part. So the process we're going to discuss is going to have to start very definitely right there and end very definitely right there. Start, continue, and stop. We can't be that simple, though. So the starting is more technically defined as initiation. So let's put that up there. So it turns out that there's a special sequence of DNA bases. You may have heard this term. It's called the promoter. And the promoter was named because it promotes transcription. What it does is it attracts RNA polymerase and orients it in the perfect way on the double helix, so that it's, when it starts to polymerize, it's starting at the exact right spot. I think you can appreciate that there's no wiggle room here. Transcription must start with so-called +1, the first nucleotide in the gene. If it misses that or starts too early, everything's going to be ruined. So this is a very definite and no margin for error type of situation. Now, I don't know how much detail your course is going to go into about the promoter, so I'm going into an intermediate level of understanding. It is a DNA sequence, and there are 2 particular regions in bacteria that are important with respect to the promoter. I say that it's an upstream region, so let me show you what I mean by that. Let's again have a piece of DNA. Let's say that +1 of the gene starts there. Okay? You could put the polarities in here, but maybe we should. If this was the template strand, see, RNA polymerase, we're going to say it right now. Like DNA polymerase, RNA grows in the 5' to 3' direction. So the template strand is always read in the 3' to 5' direction. So if that's the template strand, let's say we have 3', and I'll just pick a random spot there, 5'. The other strand would be 5' to 3', right? So we're going to start copying here, and this blue line is representing the new RNA. The promoter upstream means in this position, okay? On my diagram to the left, I mean, no, DNA doesn't know right and left, but if you take into account the polarities that are there, the promoter would be located somewhere in this region over here. So you can picture RNA polymerase coming right over here, getting ready and getting set, and then going in that direction, starting right exactly with the +1. Now, -10 and -35 regions, it turns out that those are very definite places where RNA polymerase contacts the DNA, and there have to be rather specific sequences there in order for that to work correctly. So this is a very highly orchestrated event. The -10 and -35 regions are most important for prokaryotic promoters. Now this is why the sigma factor is special. Once RNA polymerase is situated there, the sigma factor dissociates. So what that tells you is that that's not necessary for in our simple version. It's necessary for the starting, but it's not necessary for the continuing and the stopping. Okay? So initiation, starting, the promoter has to get RNA polymerase to go to the right spot. Second is elongation. So as I just mentioned, RNA polymerase will add nucleotides always in the 5 to 3 direction, because in that sense RNA polymerase is like DNA polymerase. It only can, you can imagine why. I don't think I need to say it again, because with respect to the 5' and 3' part of the nucleotide, it's pretty much the same in DNA and RNA. Those are the places where the polymerization has to occur. And the substrates here are NTPs. I forget if we used that term, but it just means one of the 4 triphosphate nucleotides of RNA. So whichever one's called for according to the base pairing rules, right? So basically you picture it started, and now it's going to be shooting along, and it's going to keep going until it's told to stop. And that stopping has to be at a very specific place as well. So stopping is technically called termination, and it's sort of the opposite of the promoter, a DNA sequence element called the terminator, not that guy in the movie, but the terminator causes termination. So it causes, for reasons we'll see in a minute, RNA polymerase to stop and get off, which causes chain termination, and there's your RNA molecule. Now, messenger RNAs in particular can range from, very short, like 50 nucleotides, to thousands and thousands. The only way it knows is it knows because the promoter tells it to start, the terminator tells it to stop. So you end up with a piece of RNA, again, usually equivalent to 1 gene's worth of DNA. All right? So we have initiation, elongation, and termination. Now here's the picture that I've been wanting to get to because I really like it a lot. I hope it's going to make sense. There are similarities with DNA replication, but there are also differences, right? So just to recap, the similarity with DNA replication is that a DNA strand is being used as a template, and the base pairing rules are being used to see what new nucleotide should come over, if you will. So this over here, I really didn't mean to do that. I'm sorry. I wanted to make a nice circle. The DNA template strand in this case is going to be this one here, right? Forget about the other one just for a second. We want to see the similarities. So we know if there's a T there, A and T are complementary, so an A is going to be there. And if there's a C there, there's going to be a G here, and et cetera, right? So in that sense, with the exception, of course, that when there's an A here, U is going to come over if we're building RNA. But the idea of lining up these nucleotides according to the base pairing rules is similar to what goes on when DNA replicates itself. One difference is, though, look, the coding strand or non-template strand is just hanging out here really doing nothing, right? Literally, it's getting out of the way. Now, it's hard to visualize in this picture. I believe we do have an animation that you could take a look at if this doesn't work for you. But these arrows here are meant to show you that we don't call it a replication fork, but RNA polymerase, which is this red sort of oval here, is proceeding in this direction of these arrows. So what that means, guys, is that the promoter was somewhere down here, right? The terminator is going to be somewhere up here. And in the middle are the nucleotides that we wish to make a copy of. Now what happens is RNA polymerase also is fancy because as it's going, it unwinds little pieces of the DNA helix that it's working on. And what that creates is a bubble, a transcription bubble, that appears as if it's moving. Because once it's done, look down here, this part is already dissociated off the DNA, and the DNA down here has folded back up into its double helical configuration. So once RNA polymerase is done with a certain part of the template, it refolds and it's opening up new parts of the template as it progresses up or down the DNA molecule, right? So it's progressing this way, and it's going to unfold this area next and just keep going until it's told to stop, right? I want to erase this so we can just make sure we have an idea about the relationship here between the template and the coding strand. Look why it's called coding. My drawing before was terrible. This is a lot better, obviously. Here's the coding strand. So look, what is the sequence here? A, G, C, T. Is that not the same exact sequence of the RNA that's just been made in the same vicinity? A, G, C, well, there's a U there, but U basically equals T in the RNA world, right? So my point before was, if you want to read the sequence, the coding strand has the same sequence or the similar sequence as the newly made RNA. The template strand has the complementary sequence because it's acting as the template, right? So you could follow it all the way down. I'll just do a couple of them. If you want to even look down here, for example, you'll see it got twisted around, but that A, that C, U is always replacing T. So the RNA is, this is another, it's not another way, it's further evidence of the way that this is somewhat similar to DNA replication. One way that it's definitely not similar is that only one strand is being used as a template. And another way that it's definitely not similar is that we're only copying a very small region of this molecule, right? One gene. All right, so the promoter, we said, is the place where RNA polymerase is attracted to. It might be a little more difficult to understand the terminator and why termination occurs. It turns out in bacteria that there are two types of termination. Rho dependent is a little easier to understand, I think. It's not actually the most common way, but let's just mention it. There's a protein called Rho. It's the Greek letter Rho, which is like an R. It recognizes a termination sequence, and it binds to that termination sequence, this Rho. So you can think of RNA polymerase motoring along, and all of a sudden, it bumps into Rho, and it sort of gets knocked off the DNA. It's hard to think of these things at the molecular level, but that's what's going on. So it's one way to stop transcription is to have a protein bind to the terminator and create a physical barrier to the progression of RNA polymerase. More commonly, and this does illustrate another point that we really want to make sure we understand, Rho independent, so you don't need a protein here. Here, this is very interesting. We haven't mentioned this before. We've said RNA is single-stranded, but there's a little asterisk to that. A Rho-independent termination sequence is a GC-rich stretch of the gene on the template strand, followed by 8 to 10 A's on the template strand. Remember that GC bonds are a lot stronger than AT or AU bonds for that matter, okay? So even though RNA is single-stranded, if it's allowed to, it very often will fold back on itself to make various secondary structures that turn out to be actually very important for its function in a variety of ways. It's probably beyond the scope of this course, or at least this discussion right now. But let's just look at one example of that. So what this allows the formation of is something that's referred to as a hairpin loop secondary structure, or some people call it a stem loop structure. It's a little hard to visualize this, but it tugs the nascent mRNA, grabs it right off the DNA template. But grabbing implies something else doing it. It just sort of happens. It does it itself. But let me explain why. Let me draw you a little bad picture, and then I'll show you a good picture. What happens is, here's the RNA, and this is one molecule that's single-stranded. But when a hairpin loop forms, there are complementary bases here, G, C, G. I can't fit them in there, but think of all GCs here being complementary to each other. That creates the stem, and here's the loop up here, and basically this is on the newly made RNA. Now if there are a lot of A's on the template strand, there are going to be a lot of U's over here, right? And what happens is this is such a stable structure. You have to think of it this way. when the RNA comes off, it snaps into this structure and basically tugs that stretch of views off of the template because they're weaker bonds. Let's look at a proper picture of that. So here is, you can see, this is the relevant part of the structure here, is all the C's and G's. In this case, it really doesn't matter which is on which side. The point is to make a very stable bonding here. And interestingly, this is a little double helix. I can't probably draw it that well, but it's like that little part of the molecule, if it's paired like this, is in a helical configuration. The loop is hanging out there. And then picture this, it's being made, right? First this gets made, then the loop, then this gets made. When it's done, that snaps together and it pulls this part off because all these UA bonds are not that strong, and the snapping together of the hairpin causes termination. You may think that it's a little bit hard to visualize, but that's what happens. And just as importantly, we've now learned something new about RNA, is that its secondary structure is not only possible, but very relevant in some ways to its function. So even though it's single-stranded, we will often see RNA binding either with itself or with another RNA molecule, or even with another DNA molecule sometimes. And the base pairing rules are always the same, so that they're what governs these type of structures being formed. Okay, so a couple of other points about transcription. We didn't get to translation yet, where the messenger RNA is actually used to make the protein, but we know that it exists. And in bacteria, I just wanted to get at the idea that transcription and translation can occur at the same time. Literally, they're both going on. So the reason may be obvious for this, since the DNA is not in any nucleus. There is no nucleus, right? Everything's accessible in the cell. There's no boundary between where the DNA is and where the ribosomes are. So as soon as a piece of messenger RNA starts getting made, it can attach to ribosomes. I know we didn't talk about that part yet, but just be aware that these two processes happen simultaneously in bacteria. This could never happen in eukaryotes because, as we're going to see, an RNA molecule has to fully be done, leave the nucleus, and go out and find a ribosome before it can begin being translated. So I think I just said this, but let's make sure. Translation can begin before the transcript, which is, I'm sorry if I didn't mention this, a messenger RNA is often called the transcript. It is the copy of the DNA, the relevant part. So before it's even synthesized fully, transcription doesn't have to be done before translation can start. And I guess you understand why this could never be the case in eukaryotes. So it's not that it's not important, it's just hard to know how important it's going to be according to your professor, let's put it that way. Now, informationally, we've been focusing on messenger RNAs. And you probably know that there are other types of RNAs around, but all of them are produced in basically the same way by transcription. So the messenger RNA we've been focusing on, or mRNA, that's one of the reasons we're focusing on it is because that's the information from the gene that's going to be used directly to make a protein. tRNA, or transfer RNA, we'll talk more about it, but I call this the decoder molecule. It functions in translation specifically by bringing the right amino acid over to the ribosome when it's directed to do so. But the decoder idea comes from the idea that it speaks both languages, both amino acid language and nucleotide language. rRNA, or ribosomal RNA, it turns out that there are three very definite types in bacteria of three different sizes. There's 23S, 16S, and 5S. What the heck is S? Well, S is a sedimentation coefficient. All this means is that in the early days, and sometimes even now, the way large molecules get separated is in a centrifugation gradient. So larger molecules are going to be going faster and further down the gradient than smaller ones. So I think S actually is named after a scientist named Svedberg. I think it's a Svedberg unit, but it basically means the larger the S, the bigger and denser the molecule. That's it. So 23S RNA is a lot bigger than 5S RNA. But these are three very definite pieces of ribosomal RNA, and there's about 30 or 40 ribosomal proteins. And the ribosome is gigantic if you think of it as compared to just like a little protein or RNA molecule. As a cellular organelle, it's not particularly large. It's a macromolecular assembly with three relatively large RNAs complexed with a bunch of different proteins. There's evidence that some of the ribosomal RNA is catalytic, read enzymatic, but right now we're not going to go into that too much. It turns out to be rather interesting and important, but we'll talk about that a little bit later. Okay? So those are the three major types of RNAs. Again, I told you that I like bacteria and that all of this was discovered in bacteria. So far our discussion has really been about the process in bacteria. Conceptually, it really happens very much the same in eukaryotes, but there are a couple of really fundamental differences, I guess. I mean, they're not fundamental in terms of information flow, but it's just there are certain things because of the way eukaryotic cells are arranged that have to be different. And, you know, eukaryotes are like Texas. Everything's bigger and better in them. I don't really mean better, but you know what I mean. Things are more complicated. They're, because of the compartmentalization, etc., you know, they're larger cells. So anyway, let's just look at some basic differences between what we've said and eukaryotes. Again, sorry to keep saying this, but I can't know how much it's going to be emphasized in your particular class. But aside from the three classes that we just mentioned, in other words, eukaryotes do have rRNA, tRNA, and mRNA, they also have another group called snRNAs, small nuclear RNAs. And they function in RNA splicing, which we're going to talk about in the next session. But RNA splicing doesn't occur in bacteria, it only occurs in eukaryotes, so that's what the small nuclear RNAs are. And they're small and they're in the nucleus. Makes sense, right? Also, there are three very distinct and different RNA polymerases present in eukaryotes, abbreviated RNA pol 1, 2, and 3. I think they were named according to the discovery date of them, so the number, you have to know it probably, but it doesn't really mean anything. Pol 2 is the one that transcribes messenger RNAs, okay? Pol 1 transcribes the ribosomal RNAs. Now, I think this will make sense in this idea that in bacteria we just saw a 23, a 16, and a 5S, right? The eukaryotic equivalents are very similar. They're just a little bit larger, so they're 28S, 18S, and 5.8S. But those are the three basic ribosomal RNAs. Polymerase 3 transcribes the transfer RNAs and also makes a 5S ribosomal RNA. This could get a little confusing because there is a 5S RNA in bacteria, but it's equivalent to that 5.8S one. There is no bacterial equivalent to the eukaryotic 5S RNA. It has a special function. We really don't need to get into it right now, but there are 4 ribosomal RNAs technically in eukaryotes. Also, polymerase 3 makes most of the small nuclear RNAs, so it seems like Pol 3 is, aside from the transfer RNAs, it's also sort of making the special or different RNAs. Pol 3 genes often have an internal promoter, which is very, very interesting. We don't need to go into it in ridiculous detail, but suffice it to say that if this is +1, mostly we've said that the promoter would be over here, right? With Pol 3 genes, the promoter can actually be located within the transcribed sequence. It's not easy to understand how this makes sense either physically or informationally, but it's a good thing to keep in mind. All right? There are probably a lot of other much more specific differences that we could mention. In terms of the process, again, fundamentally it's the same, but here are some differences you might want to keep in mind. So there is a promoter, but the promoter is more complicated in eukaryotes, as you probably might guess. Instead of a -10 and a -35 region, there are in particular three specialized regions that are necessary that exist around -30, -75, and -90. An AT-rich area that's called the Tata box, which may sound a little silly. A box that contains the sequence CAAT almost always is the CAT box, and the GC box is a GC-rich area. There may be there are additional upstream elements as well. My point is that you can't make a generalized statement about them here. Now, this can get very, very complicated. I don't know how complicated you're going to get, but in bacteria, RNA polymerase can bind right to the promoter without any additional proteins being present. In eukaryotes, other proteins called transcription factors are necessary to be bound to the DNA to interact with RNA polymerase to allow it to bind in a stable and correct orientation. And this whole complex of RNA polymerase, transcription factors, and the DNA is referred to as the initiation complex. Now, this statement, you know what? I'm going to skip down to this one just for a minute. There are elements called enhancers that are definitely unique to eukaryotes. Enhancers enhance transcription. They're not the same as promoters because promoters interact in a very definite way with RNA polymerase. So they're called enhancers, but they're interesting because their location is not necessarily predictable. They may be located far away, pretty far, hundreds of base pairs from the promoter, and yet they enhance the activity of a particular gene. Bacteria definitely don't have that. Now this one I wanted to save for the last on this slide because there are regulatory elements which determine whether or not a gene will be expressed, but hear me out for a second. In bacteria, there are also regulatory elements. In other words, genes always have to have the ability to be turned on and off. That happens in both eukaryotes and prokaryotes in a general way. But what I mean here is, if you're talking about a eukaryote that's a multicellular creature, especially an animal like yourself, there are regulatory elements that have no bacterial equivalent, that are tissue specific, for example. So without going into a ridiculous long discussion about it, all the DNA in all the cells of your body, barring mutation, is the same, right? And yet you have eyes and a nose and a big toe. So tissue-specific regulatory elements basically tell genes in your liver, hey, you're in the liver, you're supposed to be on, but they're not going to be on in your eyeball. Whereas the ones that are your eye genes have specific elements that say, okay, turn on here. Obviously, a bacterium doesn't face that problem, right? It faces different problems. But those are the type of regulatory elements I'm saying that are unique to multicellular eukaryotes anyway. All right, well, that's quite a bit to take in, I think. So let it seep into your mind, take a little break, and in the next session, we're going to continue talking about RNA molecules and talk about some other structural and functional aspects of them, as well as how they have to be processed in eukaryotes. I'll see you then.